Hi everyone, we will discuss translation chapter in this one. We have discussed DNA, we have discussed replication. Now we are discussing translation. What is translation? Sorry. Transcription. We'll discuss transcription. We've seen that the DNA structure and how it is replicates. Then we'll see how it is transcribed to RNA. You know that central dogma where the information present in the DNA transcribed to RNA, then transcribed to protein. That everyone we know. You know this procedure. But how it happens? This happens through transcription, DNA to RNA, and RNA to protein happens through translation. We will discuss transcription and translation together. Then what is transcription? It is just the process of synthesizing RNA from the DNA template. It is the first step of gene expression. It occurs in both prokaryotes and eukaryotes. And we will discuss how it occurs in prokaryotes and how it occurs in the eukaryotes. What's the difference between the transcription of the prokaryotes and the difference of eukaryotes? And which is a major key player in the transcription is your RNA polymerase. It actually catalyzes the transcription process by copying one of the strand DNA into RNA. Means it adds the ribonucleotides as a complementary strand, then it forms the RNA. What different types of RNA is going to produce? Like messenger RNA, where it is majorly for gene expression, the tRNA. Okay. We know that function of tRNA many times asked, brings the amino acid during translation. Then rRNA, it's mainly important in translation. This where your protein synthesis will occur. And SI, small nucleotide RNA, we discussed in detail. Okay, small nuclear RNA, majorly involved in the splicing. Then miRNA, siRNA, we have discussed how it regulates the gene expression. These are the different RNAs are produced. Then, what are the different steps in the transcription? The three major steps, they are initiation, elongation, and termination. What happens in the initiation? First, your key player RNA polymerase will bind to the 3' site and start producing RNA from 5' to 3' direction. Then there is elongation, growing of your RNA chain. Then at particular point, the process will terminate. And there will be formation of your 5' to 3' RNA. This again go for translation and your gene is going to express and trait will going to express. This is how actually the transcription occurs. Then we look into one by one what is happens in the initiation. If we take prokaryotic transcription initiation, this it is I am discussing now prokaryotes. Please remember. One of the main important enzyme in prokaryotic transcription is RNA polymerase enzyme. It is a hollow enzyme. It consists of two things. One is core enzyme and sigma factor. If it is no sigma factor, it's only core enzyme. Core enzyme consists alpha 2, beta dash, omega complexes. It actually catalyzes the RNA synthesis. So its elongation is very important. Where sigma factor, actually there is one saying, the RNA polymerase without sigma factor, it's like a street vendor. Like I don't having any, I don't like how stranger will move around the village without address. Like if you don't have address, you are like a stranger. Like simply you're wandering. You don't know where to go if you don't have the address. Same way, without sigma factor, it's a like wizard wanderer, some one quote comes. Actually, it is called as like in a metaphoric word. They used to do use that word. The sigma factor with core enzyme can't able to bind or can't able to locate where it needs to be bind. The sigma factor it actually recognizes the promoter sequence and it tells the core enzyme, okay, you have to bind here in the promoter region. Okay? That is the very important function of sigma factor. Many times asked in the exam. What is the role of sigma factor in the transcription? Means it guides your core enzyme to bind at the promoter site. Then what is the promoter site? It's very important. We will discuss. Promoter sites having two major sequences, start site and 10 -10 sequences and -35 sequences, where -35 sequence where sigma factor will recognizes this sequence and binds. Then this 10 -10 is also called as Pribno box, like TATAAT. It is a consensus sequence. Should know what is the difference between consensus sequence and conserved sequence. It's there in the medicine. Please read it. Okay, I'm telling read it. I'm not telling here. Because if you tell here, you're not going to read. Then if you told here, you're not going to read it. Then what is it's a consensus sequence where your RNA polymerase will bind and start unbinding. So here, -10 sequence, where the recognition happens -35 by sequences. Then what are the different steps? First, there will formation of closed complex where RNA polymerase binds to the promoter. Then there will be unwinding. See, there is an unwinding of your DNA strand, then formation of open complex. Then there is a formation of short RNA sequences. Like this, there will be formation of there will be short RNA sequences like 2 to 10 nucleotide. But again, this transcription stops. Again, there will be formation of 2 to 10 nucleotides of RNA. Then again, transcription stops. Means it's a like it is called as abortive transcription. Okay? Means short RNA. Then it is actually not a real transcription. First, it is going for abortive transcription where your short RNA molecules were raised. Means it's a preparatory step. Then after that, after promoter clearance, means RNA polymerase, once it's completed all the steps, unwinding and abortive transcription, it moves forward. From here, see here it started initiation, it has to move here. This movement is called as clearance. Means where after recognition and after initiation, the sigma factor released from promoter. Sigma factor released after initiation. Means in elongation, there is no role of sigma factor. Please remember, many times asked in the exam. What is the role of sigma factor in elongation? No role. But only in the promoter recognition, it having the role. After completion of initiation, promoter clearance, sigma factor will released. Okay, this point is very important. Means RNA polymerase move forward along with the chain, then releasing of sigma factor. Then how eukaryotic transcription will occur? Here there are several RNA polymerase 2, RNA polymerase 3, but we will just look into the RNA polymerase 2 promoter. It having Tata box having -25 to 30 base pair, and where it's having the RNA polymerase binds Tata binding promoter. Then we having BRE sequences like -35 to 37, and we having initiator sequences, and we have proximal promoter sequence that we will discuss in detail. They have different promoters, like not only eukaryotes, in prokaryotes also possible. See, in polymerase 1 promoter, it's having upstream element like 200 to 10 -10 sequence and having core element sequence. In RNA polymerase 2, it having -100 base pair sequence, -80 sequences called as cat box, and GC box that is called as proximal control elements. Then another one is core promoter. Please remember what components comes under core promoter, what components comes under proximal control elements. You should remember. In proximal control elements, we have GC box and cat box. And in core promoter, we having Tata box and start point like intron, sorry, initiator sequences. Then we having your coding region. Then we having termination site. For RNA polymerase 3, we having tRNA genes. Like for a tRNA, there is separate promoter like A box and B box. A box from 18 to 20 and B box 50 to 61. Like 5S ribosomal RNA gene, yeah, it is also replicated by RNA polymerase 3. Please remember, RNA polymerase 3 replicates both tRNA and 5S rRNA. Normally, generally we think 5S rRNA also by RNA polymerase 1, but it's not. RNA polymerase 1 not synthesizes your 5S rRNA gene, where RNA polymerase 3 actually synthesizes 5S rRNA gene. Okay. Then, like, what are the different polymerases in eukaryotes? RNA polymerase 1 actually it is located in nucleolus. It forms ribosomal RNAs, but it not synthesizes your 5S rRNA. RNA polymerase 2 it's present in nucleus, where mRNA, majorly mRNA or miRNA is produced by this one. And RNA polymerase 3, tRNA, 5S RNA, other small nuclear RNA like involved in this splicing is created by RNA polymerase 3. There are others present in plant, RNA polymerase 4 and RNA polymerase 5. They are producing some small interfering RNA, siRNA that we discussed, how it is regulates the gene expression. And some antisense target like siRNA target genes also produce. Remember, RNA 4 and 5 is present only in the plants, but rest all these present in the all the organisms. RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. Then another unique feature in the eukaryotes like RNA polymerase is having C-terminal domain. What is this? It's a continuous stretch of amino acids. Actually, phosphorylation and disphosphorylation of phosphorylation, phosphorylation and disphosphorylation actually regulates the transcription. Based on the phosphorylation, actually it activates the RNA polymerase and unwinding will happen. That's why the C-terminal conserved domain of RNA polymerase is very, very important in the regulation of transcription. That what are the different steps in the eukaryotic transcription? First, formation of PAC complex means pre-initiation complex. Then first what happens, there are different transcription factors which binds to your DNA. Then there is a formation of pre-initiation complex. I'm not discussing initiation complex, but this was unique feature of eukaryotes. These transcription factors were not present in the prokaryotes. Then what are the different transcription factor? There are transcription factor D, transcription factor A and B, transcription factor F, RNA polymerase, and transcription factor E and H. We'll see what is the function of each. Transcription factor D actually first transcription factor which binds to the DNA like Tata binding protein, like a Tata box. Then A and B will come, stabilizes this complex. Then transcription factor F, which brings the RNA polymerase. This one is very important, and D also very important. Which transcription binds first time is transcription factor D, and which brings the RNA polymerase is F. Then E and actually H recruits this transcription factor F activity. Then there is a formation of open complex, means transcription factor 2H, which unwinds your DNA. Then after that, there will be clearance of the promoter where your CTT domain of RNA polymerase is phosphorylated and it will start elongation. In elongation, what will happen? Where the 5' to 3' direction, your RNA start growing, RNA chain start synthesizing, where A is complementary for A, complementary U is added, for T, complementary A is added, rest other the like DNA replication. Then major elongation factors in prokaryotes are NUS-A, where it prevents the premature termination, and in eukaryotes there is a TFB protein which phosphorylates the CTD domain. We have discussed what is CTD domain. Then here also there is a supercoiling of DNA. You have to remove the supercoiling that is done by topoisomerase enzyme, either gyrase or topoisomerase 1. Please remember this one. and this is called as transcription bubble. It having like 18 different books and different organisms different. Normally it is having 10 to 20 nucleotide bubble, nucleotide sequence there in the each transcription bubble. It's open complex having 10 to 20 nucleotides. It's called as transcription bubble, wherever DNA synthesis, like RNA synthesis will occurs. Then how termination will occur? Actually, there will in two types of term. In prokaryotes, we having two types of termination. One is intrinsic termination and row dependent termination. We will discuss intrinsic means where RNA forms GC-rich hairpin loop followed by U-rich region. It causes RNA polymerase to pause and dissociate. In row dependent, what will happen? There is a specific protein called as row protein. It's a ATP dependent helicase, binds to rut site in RNA and moves forward to RNA polymerase and disrupt the complex. We will discuss. For example, intrinsic, as I told, there is a RNA forms GC hairpin loop followed by U-rich region. See why there is in DNA there are some regions having inverted repeat sequence followed by A. When after transcription, what will happen? This transcription will happen. What happens? There will be this inverted sequence. They will anneal. There is a formation of this hairpin loop followed by U-rich region because A is a U-rich region. Because of this hairpin loop, RNA polymerase can't continue this chain. That's why RNA polymerase will dissociate from this complex and your termination will happen. In row dependent, it's a different mechanism. In RNA, this one having some rut site where row binding protein will bind here. After that, your RNA polymerase will not take place. This is called as row dependent termination. You should know what is the properties of row protein. It's a DNA dependent, sorry, ATP dependent DNA helicase enzyme. Sorry, helicase properties. Then one of the another unique feature of the transcription of this from prokaryotes. In prokaryotes, what happens? Your transcription and translation occur together. Transcription and translation occur together because it occurs. All things are occurring in cytoplasm. After DNA replication, your transcription and translation occur together. But in case of your eukaryotes, transcription occurs in the nucleus, then it transport into the cytoplasm, then translation occur in the cytoplasm. It occur at the different times at different location. But in the case of both transcription and translation occur at the same time. This is one of the unique feature of the prokaryotes. Then in eukaryotes, how termination will occur? Either through poly A dependent termination or RNA polymerase 1 type termination, RNA polymerase 3 type termination. In what is happens in poly A dependent termination, where RNA polymerase 2 passes polyadenation signals. For example, at 3' end, there is a one sequence is called as AAA, UAA. It actually, this is a signal sequence for termination. After when RNA polymerase passes through this sequence, there is one particular enzyme called as endonuclease, CPSF enzyme, which cleaves the RNA at the downstream. After cleaving, what will happen? It adds the 100 to 250 poly A tail to the RNA chain that we will discuss polyadenylation. Then the termination will happen. It means that termination is actually happens when it's not like a complete synthesis of RNA after termination. It's not like that. It's like a co-transcription. When transcription is still happening, this termination will happen. Then after that, RNA polymerase to is phosphorylated and dissociates. Then RNA in another type, RNA polymerase 1 type, actually there is a specific protein called TTF protein, which actually causes your termination of rRNA genes. Then RNA polymerase 3 like tRNA and siRNA genes, actually it's like your prokaryotic intrinsic termination. There is a formation of hairpin loop followed by U-rich region. Then your RNA polymerase dissociate and causes termination. This is how termination occurs at different types of RNA in eukaryotes. Then you have studied what is transcription and your RNA is formed, but it is highly unstable. We need to do some processing so that we have to make it stable. How the different processing will happen? there are three different major processing. One is capping, and another one is tailing, and another one is splicing, and another one is editing, RNA editing. These four mechanisms are very, very important that we are going to discuss today. What is capping means? Addition of 7-methyl guanosine cap at 5' end. And one of the unique feature is 5' to 5' sequence. See, 5' to 5'. This is asked many times in the exam, where this 5' to 5' sequence is present in the organism, where your capping occurs. Here 5' to 5' sequence occurs. See, there is an addition of 7-methyl guanosine to this region, the 5' end of the mRNA. It is called as capping. Whereas in 2023, there is one question was asked, like trimethylation, monomethylation, dimethylation. Normally, monomethylation is more uh, predominant in eukaryotes. That you should remember. Cap 0, cap 1, cap 2 were there. You just read about that. Okay. Then, what is the main function? It prevents the degradation of RNA, and it also capping is very, very important in translation initiation. Like we'll discuss like how initiation of translation happens by using 5' cap. We will discuss in translation chapter. And you understood, I think, adding of 7-methyl guanosine to the 5' end of the mRNA, and the 5' to 5' pairing is unique feature of this capping. You should remember these points. And what is the importance of capping is prevents the degradation and enhances the translation initiation process. Then another one is splicing. Means here it's simple, in a simple words, its introns are removed and exons are joined. There are different types of splicing we will discuss, but one of the major important key player in splicing is spliceosome complex. It is actually complex of small nuclear ribonuclease proteins like U1, U2, U4, E5, U6, which removes the intron. How it actually happens? First, before going to that, we have to discuss what is the splice sites. There are normally AG-GT splice sites, like GU-AG splice sites, a 5' splice sites, and this is 3' splice sites, where actually your splicing will occur. Okay. Then, for example, here intron is there, here branch point is there, 5' splice site and 3' splice site. Every part is very important in splicing. We will discuss that one. Before that, coming to that, we'll discuss small transcription, like splicing action in tRNA and ribosomal RNA. In splicing of tRNA, majorly occurs by one of the endonuclease enzyme, which cuts the DNA at particular. Here intron was there. Here there will be endonuclease cleavage will happen. Then removal of this intron. Then there will formation of mature tRNA. It is just a simple procedure where endonuclease will cut and joins, then mature tRNA will forms. But in rRNA, there is autolytic splicing. Means there is no requirement of any spliceosome. There is no requirement of any snRNPs for splicing of your rRNA gene. There are group 1 and group 2 splicing that I am not going in detail, but you should remember there is a self-splicing occurs in the rRNA. There is no need of all these snRNPs for the splicing of introns in the ribosomal RNA. That there is a formation of Lariat structure. There will be splice site inside this one. It will protonated. There formation of Lariat structure, removal of Lariat structure. That we will read if you want to. If you are interested, read this in the Pranav Kumar book. What is Group 1 intron splicing, Group 2 intron splicing in rRNA? It's very interesting, but for SR, it is for CSR, it's very important. For SR, not that much important. But you should know in ribosomal RNA, autoclatic Group 1, Group 2 mechanisms were there. Then in splicing tRNA, just endonuclease cleavage. Then in mRNA, how it happens? It is mediated through spliceome-mediated splicing. We having, as we discussed, there are several proteins like U1, U2, U4, E5, 6. They are actually control the splicing. And this question in this 2024 exam, they are asked U4 and which one U4 and U6 always together? Sorry, in CSI it asked. Okay. Then how actually it occurs? First, First, as I told, there will be exon and there is an intron having branch site, and there are several times exon definition. I'm not going in detail, exon definition, intron definition, it's all not required. Okay. Here, what will happen? GU splice site and AG 3' splice site and 5' splice site, and there is a branch site where UN binds to 5' splice site. Please remember where UN will going to bind, and U2 bind to branch site. Then after that it recruits all complete this one U1, U2, U5, U4, U6. Then there is a at 5' end the introns are joined to A, the branch side. This intron will come and branch to A. Then U1 and U4 will going to release. Then after that there is a complete removal of this one intron region, then joining of exons. This is a very simple procedure. You just remember for SR, they're not going to ask in detail. What is spliceosome complex? snRNA is very important. What are the different types of snRNP proteins were there, and where actually GUAG splice sites were there, and what exactly it will how exactly it joins the exon and how remove. And this structure is called as Lariat structure. If you remember, this much is more than enough for splicing. Then after splicing, no, not after splicing, it is another processing mechanism, polyadenylation, where at 3' end of the RNA, we are adding 100 to 250 adenines to the RNA at 3' splice, sorry, 3' end. What is the function in this one? Stabilizes the mRNA, enhances the nuclear export, and regulates the transcription efficiency. For example, if RNA is not polyadenylated, it remains in the nucleus only. It will never come outside of the, sometimes it will come, but generally it will not come outside of the nucleus. Only after proper processing steps happens like capping and terminate, like polyadenylation, then only it will come outside. If it is simply come outside, it will degrade it. That's why capping and tailing is very, very important. Then, like, see your at 3' end, there is addition of your poly a tail, then your tailing will occur. Then another processing mechanism is RNA editing mechanism. This RNA editing is nothing but either we can edit the RNA by changing the individual bases or by adding or deleting some uridine residues. There are, like, for example, there are some classical example is called as apolipoprotein. If, for example, unedited RNA you take, they having 4563 amino acid long chain protein will come. But if you edit a single base, see here instead of CAA, here you are editing. See, instead of C, you are giving UAA. What is the function of UAA? It's a stop codon. See, this portion will not going to translate. Then there is a formation of only 2153 polypeptide chain. See how much difference? Half of the polypeptide will not translated because of single mutation from C to A. This is called as the editing of your RNA. It can also occur through another thing like guide RNA mediated RNA editing. We provide some guide RNA. We discussed how CRISPR RNA, like in the same way, guide RNA which guides where your editing should happen, then your RNA editing will happen. This is how you can edit the RNA also. This is the, in summary, capping, splicing, termination, and RNA editing. So major 4 mechanisms for post-transcription modifications. Okay. Then how actually it is regulated, how transcription is regulated. There are several model, operon model, lac operon, tryptophan operon model that we will discuss in the next session. Then in summary, there are in eukaryotic there are some enhancers and silencers. In enhancer sequence, actually it binds to activators, then increases the transcription. Many times it asks, what is the property of enhancer? Transcription factors and all those things. Enhancers. Enhancer means there are several sequence, either it's near to your gene and any side. It is like either it is upstream or it is downstream. Normally, most of the promoter which is upstream to the like up at 5 prime type, 5 prime region. But enhancer it is independent orientation. Please remember, enhancer is having independent orientation. Either upstream or downstream, but it regulates your transcript. And you should remember, and it can distance also. Promoter means it is exactly adjacent to your coding region. Then only it called as promoter. But enhancer is away from anywhere from the. For example, here enhancer is there. Some far away your gene is there. Also it regulates. That is the unique properties of enhancer. Means enhancer having it is the enhancers are orientation independent and also distance independent. At whatever the distance, it can able to regulate. But your promoters are it is orientation dependent and also it has to be present exactly adjacent to your coding region. But enhancers are orientation independent and distance also at whatever the distance can able to control the gene. Then transcription also epigenetic control we have discussed, and transcription factors, general transcription factor, specific transcription factors. Like we discussed what is the general transcription factors in detail, and there are specific transcription factors like sorry MY3 and P53. They regulates particular genes and these things. Okay. Then in summary, in prokaryotic transcription, where sigma factor binds to promoter, then RNA polymerase move along the DNA. And termination either through row independent or row independent, there is no processing. But in eukaryote, transcription factor 2D and RNA polymerase 2, it binds the promoter. Then there will be phosphorylation of CTT domain. Then addition of polyatyl for the termination. Then the processing like capping, splicing, pollination, polyadenylation will occur. Okay? This is about the transcription. So just remember this much is more than enough for SRE. Okay. Bye bye.